That's a good question. I'm not sure how much that is known, given that these are probably very rare observation in general.
Yeah, the one we collected stayed inflated the entire transit up to the surface and eventually settled in a little bit and deflated.
Yeah, those eyes are beautiful. Tina was commenting on the orange ring around the eyes.
Color coordinated with the fins.
Yes.
Well, maybe we'll get a chance to see him walk along the bottom. That's always interesting. Right. Maybe when we zoom out, we can put the lasers on again just to get a sense of his size. He's quite small.
Come on out, video. Hey, whenever you're clear.
Oh, yeah.
Yeah, he's only about 15 centimeters total.
When he takes up your whole screen, you think he's a couple feet long.
He's trying to make himself look big, that's for sure.
All right, let's come wide.
All right.
Most of the water column anglerfish that I've seen are much smaller than that. In fact, like the size of my thumb.
Yeah.
But then you see these pictures of them where they're zoomed in and people think they're humongous. They're really not.
Yeah, so we're starting to see, sorry, have to mention the rock before we lose it. We're starting to see a different kind of rock. So as we go up this slope, we're literally going into younger and younger rocks that have deposited over time and layer upon layer. So the base of the slope, we're looking at the oldest, and now we've been moving up this 30-degree overall slope, and we're seeing now younger rocks. And Pilot, if we could zoom in on the rock, please. It does not have that manganese crust. Oh, sorry, we're reorganizing on our front row.
It is lunchtime, right?
Oh my gosh.
So we're probably having front row, pilot, co-pilot, and navigator switch over.
So maybe we could use this time to introduce our chat room.
Oh, yeah. We have Iris Sampalo, Kate Rose, Ken Sulak from the U.S. Geological Survey, Les Watling, and Matt Dornbach, Megan Culler, and Tina Moldovsa. I'm sure I didn't say that correctly. Sorry, Tina, I'm still working on that. Molodsova. Thank you. And Tom, something like Hands Connect.
Hands Connect, yes. We have to see these phonetically. Oksana Gangule.
And Zach Prowl.
Zach Pro. Who is my graduate student.
Oh,
Shout out to Zach.
Thanks for joining us, Zach.
He's working in Peter Etnoyer's lab with several colleagues. I don't see Rachel Bassett online, but she's been helping us a lot in the past few days, and now Zach is there. And we should probably reintroduce ourselves. In the back row, we have Stephanie Bush of the Smithsonian. You want to say a little more about what you do at the Smithsonian, Stephanie?
Yeah, actually, I'm working on a project that is
sorry, video, say that again.
Looking at biodiversity of the waters all around the U.S. So East Coast, West Coast, Gulf of Mexico coast, as well as Alaska. So this is all very important for the work that I'm doing along with my colleagues at the Smithsonian.
A view on the rock to the left.
Trying to understand who's living down here, who's living on the bottom, and who's living in the water column as well.
And I'm Cheryl Morrison from the U.S. Geological Survey. I'm a research geneticist. I have a conservation genetics genomics lab back in Leetown, West Virginia. And I've been involved with deep sea research for over a dozen years. And we've been doing similar research to what Stephanie just described, started in the Gulf of Mexico and this area, the southeastern U.S., offshore region, and we've been looking at connectivity of different structure-forming habitat of corals, especially Lophelia pertusa, and whether these populations you see in different parts of the deep sea area, whether they actually are exchanging larvae and individuals between locations or not. We've also expanded our work to look at a couple of octocorals, Primnoa and Paragorgia, the bubblegum coral that we've seen several times in the last few days.
All right, pilot.
So we've just zoomed in on obviously a rock, and this is very different from all the rocks we've seen so far in this dive, and in fact on this mission, this looks like it might be either slightly coarser grain material than the mudstones we've seen. It's hard to tell even zoomed in, but certainly it has not been encrusted by this iron and manganese. So that's interesting, and we'll find out maybe when we get a rock sample how friable it is, how much it'll break apart without a crust on it. But you can certainly see it has become a home port for many organisms that have burrowed into it. So that would make me think that possibly this is an old sandstone or something that has grains that are cemented that could be dissolved by burrowing organisms, where they can use their digestive fluids to dissolve the matrix that holds together the grains and therefore burrow into what seems like solid rock. But we saw this a lot on the continental shelf edge, but again, until we really get the hand sample, we won't know. So we're going to have to grab a smaller specimen of this.
Our video looking at lasers.
Maybe the next time we take a sediment sample, and we still have our Chaunex.
And actually, Ken Sulak gave us some very interesting information. This is Chaunex coloratus, and there are several species in the genus. When this species is disturbed and bugs out, the backward-facing circular opercular openings show black and appear as eye spots, suggesting the head of a much larger fish. Oh, that's really cool. And the short lure that we were mentioning within the cavity on the head in this species might not actually be a visual lure, but instead a chemical lure. Visual lures are more typically elongate and dangled or twitched in front of the mouth, where the chemical version is held motionless within the head pit. Very interesting.
Thank you, Ken.
Yeah.
So, yeah, I'm pretty sure that is a different species from the one that we were referring to as the red-eyed gaper on the Charleston bump.
Mm-hmm.
Yep. Yep. Yeah. Yep.
And
we were doing introductions earlier, and then I got distracted
by the rock.
But did I cut you off? I didn't mean to.
No.
Our next move's probably going to be
plenty.
280.
Well, I'm Leslie Sauter. I'm at the College of Charleston in the geology department, and I've been teaching marine geology for 20 plus years. Well, I don't want to actually tell you how long.
Okay.
And in the last decade, I've been teaching seafloor mapping. So
yeah, that
this
looks
kind
good
of dive is incredible for me. You can really,
waypoint, right?
wow,
Yeah.
that's a
much steeper
face
slope
like
that
here.
we haven't seen anything like that. And you can really see how the different layers are outcropping, even though they're draped in sediment. So we're getting this stair step
bridge over.
Yeah, we'd like a ship
area.
move.
And there's where we just were with the
bearing 280
Chaunex,
degrees,
the fish.
speed decimal 2.
Okay, we'll go 20 meters.
I didn't realize we were on such a steep slope earlier.
Speed of decimal 2.
It's a good copy. Thanks, Bridge.
Yeah, our depth now is 1,297 meters.
Wow.
That looks neat.
Yeah. It's cool features. Yes, Washley, go ahead.
ROV nav bridge. Move
Yeah,
initiated.
we can do it. As long as we find a
Move initiated.
suitable rock.
Thank you, Bridge.
That's no problem. Copy that. I'll keep my eyes peeled, and if you see one too, just let me know.
So right ahead in the upper left of your screen, you'll see how one of those slabs is sliding down the slope. It's already broken off and it's starting to drape itself. But originally these are flat lying. We're looking for a small sample of it, maybe just above that. We might be able to grab one of those pieces. It's interesting that for some reason the biota don't particularly care for this substrate except those that burrow. So, wow, look at that. Oh, a geologist's dream come true.
This must be the steep,
Yeah.
steep part.
That's incredible.
But
All
there's
right.
not much fauna,
There is
so
not
you're
much
right
fauna,
that they don't
so
like this. I think it's too porous or
It
crumbly.
might be.
Or
It might be too crumbly. We have a
crinoid anyway.
crinoids like it. That's a good spot for filter feeding if you
How far
can
into
hold
that ship
on.
move?
Ship has moved
Another
Yeah, there's
crinoid.
several.
35 feet. Stand by.
So we're going to see if we can grab a sample here.
Yeah, watch it. Anything
12
on
meters.
screen that looks interesting?
Anything that you can get, the bigger the better.
Copy
I was
that.
going to say there's a little
yeah,
one
you're
there.
fine to
The
send
little
right
one
now.
would be fine. We just really
Serious
need
can see over it, I think.
representative.
Okay. Because we might sample here. So I
And
don't know if we
those
should stop
brown
Yeah.
things are
Yeah,
likely
go ahead.
sargassum, but
Bridge is RV nav.
are usually surface
Bridge over.
Can we hold current position, please?
Okay, holding.
algae that have
Watch it.
sunk
What
to the
about
bottom.
rock with the dead center?
Dead center is fine, and if the broken rock to the left,
Do we have both rock boxes?
the smaller of those two pieces,
There's only
if
a
you
small
can get
one and the
that,
right one.
but I
and
don't
the
want
left
to be
one's
too
open,
greedy.
so you could put this rock in either box.
Copy.
The small one really is a good representation of the area, so that's really what we're
RV
looking
nav,
for.
bridge. We are.
Copy that.
Over.
This one might just be a
Nav copies. Thank you.
A
tad
little big.
large.
Okay, let's just grab the small one. It'll give us room for other rocks.
Great.
And then behind the rock, on the more steep wall, there's one
all spun
pair of
out, pilot.
Anthipathes, it looks like.
Great.
And what are these organisms on the right?
I thought that they were sargassum.
I thought
that's
they were
kind
too,
of old,
but it looks like they're stalked almost. I don't. We'll have to zoom in on them. Look at the one on the lower right. He looks stalked to me.
All right, Video, you want to come in for a quick zoom before I reach out for it?
Do you want any camera right here?
I think I'm fine for now.
Okay.
Maybe when I get to the box, I might need one.
Copy.
At some point, we need to get a sediment sample too for this dive.
Copy that. Just let me know, watch leads.
All right. But that rock looks perfect.
All right, Video, whenever you're happy, you can grab it. And then which box we're going?
There is a really small one in the starboard forward. So you could do the port forward would probably be a good option.
Pilot, we can put this in the same box as the other rock.
Okay,
As
sounds
long
great.
as there's room.
Yeah. And that's starboard, right?
You go starboard up port. Yeah,
So
there's
starboard.
a small rock in the
It's
starboard. but
fairly
so
obvious
small
which
that-
is which.
Yeah, they know which is which.
So we collected a rock previously, and it was solid black, not nearly as
just
pitted
keep an
with
eye out
burrows
on-
as this,
Yep,
and
while I get
appeared
the
to be a
sample.
iron
Copy that.
manganese, either
All right, arms coming live, 3, 2, 1.
pavement itself or heavily encrusted other rock. We won't know until we get it up into the lab.
6.
This is very, very different from anything we've seen. It's much closer to looking like the sediments, although it's even brighter white. I'm thinking it's more of a calcareous
Yeah, this button's very
rock.
sticky.
the chance that it is actually a sandstone?
And
oh, dear.
Sand.
It's not very hard, is it?
It's not very lithified.
Wow.
Interesting. Well, perhaps we could scoop it.
Do you want to scoop it?
But where would we put it?
Yeah, that's a little bit of a
Okay,
problem.
well, let's keep going, and maybe we'll try another rock later.
Copy that.
Yeah, the problem with scooping a rock, for those of you listening, is that we have rock boxes that don't interfere with the bio boxes. And the rock boxes are not totally enclosed. One of them has a lid on it, but it allows water to stream through it. So if we put a rock like this into the rock box, it would probably just disintegrate on ascent.
Let's take a peek above us.
But if we end up with some space in a bio box, maybe we could get a scoop of one and get some sediment as well.
You come up.
So Les Watling chatted here that he thinks this is part of a large ancient platform, perhaps Miocene or so in age.
Yeah, the Blake Plateau itself is a very large carbonate platform. That's why I was saying I thought it was probably carbonate. I'm glad you mentioned that so I can talk about it a little bit. Mm-hmm. The continental margin of this region is very unlike even what lies off the coast of North Carolina. North Carolina, we have a shelf, a slope, and a rise, and they kind of stair-step one giant stair. Here we have a shelf and a narrow drop-off that we call the upper slope.
Wow.
Oh, my goodness.
Wow, look at that Bathypathes. That's huge. I wonder if that's a Thuarella again in the center, the white coral. But yeah, that's a gorgeous Bathypathes.
And a nice anemone underneath it, too.
Yeah,
and now we're coming back into the other manganese iron encrusted rocks. So we'll see if they fall apart as well. But this edge that we're looking at, it's called the Blake Escarpment. And an escarpment is a rather sharp feature on the large scale. And it is the edge of the Blake Plateau, which, as the name implies, is very flat. And the plateau is constructed starting at the opening of the Atlantic Ocean as a shallow carbonate platform. And as the Atlantic expanded, the platform began to sink to its current depths of about 1,000 meters. And now all of that material that had built up is made of calcium carbonate, or mostly of calcium carbonates, which are what we see in tropical settings. So it was shallow and tropical in the early days.
It looks like maybe another Analepsamia rostrata, that yellow coral on the left side. And you can see a dead patch of coral just to the bottom center there now. Probably the Selenosmilia variabilis is the species.
There's a sea star there, too.
Sea star, and that definitely looks like that Thuarella that we collected some of earlier.
Yeah, watch it. If you want zooms on anything, just let me know.
Oh, what?
Thanks, pilot.
Okay.
Cheryl, do you want to zoom in on anything? There's another sea star up on the horizon there in the center. Maybe we could look at that.
If we could look at that yellow coral that's going by, too.
On the left?
Yeah, on the left now. Thank you.
Yep. All right, video, let's go for zoom.
Yep, that looks like an Analepsamia rostrata. It's beautiful.
And Scott France just, well, what is Scott's comment, what you just said?
Yep, Analepsamia. I was looking to see whether that is on the list of species that we're looking at for connectivity work.
That is lovely. It's nice seeing the back side of it, too, just to see the branching nature of it.
Yeah, the other side would be able to see the calyx of that the polyps sit in, and you can see that little rostrum, possibly.
Are we going to take a sample here, you think?
I was just checking to see if it was on our list, and I don't see it on the list of corals for connectivity. It would be, in my experience, quite difficult to get enough individuals from any one place. So for connectivity work, you're not looking at one sample per site, but you're looking at 10 to 30 samples per site. So you kind of need to have more abundant taxa to include there.
And look what it's growing on.
Yeah, is that dead coral?
It is.
Yeah.
Very encrusted. It looked like rock.
Yeah.
So watch, this is not a sample?
Correct. This is not a sample, just a nice video.
Copy that.
We have a Stichopathes orange whip coral to the right.
I'm going to go in quick on this video.
A little black coral on the right of that.
Snap zoom it. You can come out. All right, Washley's, do you want any other zooms before I move on?
Maybe if we could just pan around one more time to get a general scene. Okay, so we have another Analypsamia rostrata there behind one of the two Bathypathes. There's a yellow octocoral that's right next to a yellow sponge.
Nice.
Okay. Okay, great, thanks. Pilot, this is watch 1.
Yep, go ahead.
Can we zoom on that yellow sponge and yellow octocoral that's on the right side of the screen now?
Yes, we can.
Thanks very much.
Video go for zoom. Copilot, you can kill lasers.
Okay, so that's some Selenismilia that's still alive there, the pink branch in the back, and then the yellow sponge is living over the framework of what this coral used to be when it was alive. And I'm sure the more we zoom, the more critters we'll see in there. Oh, it looks like there's a baby Anthomastus up there that we caught one little glimpse of. And little white tubes were probably polychaetes. Little sponges, you can see the spicules sticking out. Little baby sponges. And that might be more Anthomastus on the right.
Video come partial.
Oh, and we've got a squat lobster at the base of this.
All right, you can come back in.
And then if we could look at that coral behind this at some point.
Yes, video come partial, sorry.
Yeah, that pink octocoral is interesting. So,
all right, video come on in.
I think that this might be a Paramaricia, this orange coral. And we have a brittle star associate. Yeah, so this does look like a Paramaricia. And Paramaricia is actually one of the target species we're looking for.
Sorry, Vidi, you said left. Oh, I see it. Yeah, copy.
Pilots, this is watch 1.
Go ahead, watch 1.
Would this be something that would be possible to sample, this Paramaricia that we're looking at here?
Yeah, we could get a snippet of it.
Yeah, snippet would be all that we would need. If possible, if we could get the piece that has the brittle star on it, that would be a double bonus. But if that's taking too much or not in reach, then just a sample of the coral is the most important part.
Copy that. Copilot, do we get lasers real quick? And then video, you can go ahead and come wide as I set up.
Copilot, we got 2 samples on the port side, so leave that closed.
Copy that.
Actually, watch it. It's not that big.
Might be floating.
So yeah, I think I could get that. We'll see. All right. Go ahead and come wide, video.
Kind of going to starboard. Both starboard boxes.
Going starboard.
Yeah, ports are full and possibly floaty.
Okay.
I did hear that in the handoff. Good to know.
Ports.
Well, actually,
starboard.
Pilot,
Ready?
this is watch 1.
Go ahead.
I've just gotten news from the chat room, and this is not the species that they're looking for. So I guess we can skip this sample.
Copy. Skipping sample. All right. Anything else in the area before we get a ship move in that you'd like a view on? Just do a couple passes.
Yeah, a couple passes would be good. There's a squat lobster. Another sponge, and then we have another black coral up at the top here.
All right, I'll call one in. Bridge is RV nav.
RV nav, bridge?
Yeah, we'd like to move 20 meters, bearing 280 degrees, speed decimal 2.
Copy. Roger that. 20 meters, 280 at decimal 2.
It's a good copy.
Yes, after that. Is that trash? Yep.
Oh boy. Yeah, our first piece of trash that we've seen this dive. Yeah. It's like right above an Anthomastus.
RV nav, bridge, move initiated, over.
Move initiated. Thank you, Bridge.
Copy, I'll make my way there. Alright, video, go for zoom.
That's another cutthroat eel.
Kill lasers.
Synaphobranchus.
Copilot.
He's yawning. Maybe he's about to have a meal. Nope.
Navigator, I have a question.
Go ahead, Copilot.
Alright, video, you can come live.
So right now,
back a little.
if the ship starts drifting, it'll drift to the west because
Wow.
of the surface current, right?
It's rather vast.
Yep. In
So-
a dead ship situation. But it's a pretty small surface current.
Yeah, but let's say that it was, well,
Now it's going north.
let's say that the surface current was to the west.
Okay.
and it was larger than that.
Okay.
So we would come off bottom and line up, so as if we were streaming that way.
Yep.
Okay.
Yeah, in this situation, because if this was like
go for
steeper,
zoom video.
then you'd really have to haul in quicker, you know?
Looks like we have a shrimp in the water column. This looks like the same one we saw previously.
Copy that.
Wow,
same
it's
one.
such a
We'll
beautiful swimmer.
Just get a quick.
But yeah, if we like fell out of DP,
Yeah.
we just come ahead.
So those are
video, let's let it
they're
swim out.
pleopods,
Also, like, which way would
which
we
are
actually
underneath
line
the
up?
abdomen that are
So if the ship like loses power,
modified for
and dead
good
ship,
swimming ability.
you know, then it would line up. G2 would be facing east. So you'd line up this way, and then ship would pull you that way, and you'd haul in. Or
We're at a very steep
if we
part
just
of
lost
this
DP, then the
escarpment
ship
now.
can just push ahead.
I think we have a
D2 turns left.
yellow
you turn
sponge
right.
in the center and maybe
Or you
another
actually turn left.
Analopsamia
Yeah,
rostrata
you turn left.
in
Try to zero the
the
turns,
right
but
below that,
Yep.
close to the
Good time.
lasers.
Okay.
Do any zooms video? Or sorry, watch it.
Yeah, if we could zoom on the coral that's yellow and to the center, yeah, right there.
Yeah, video
you.
go for zoom.
Yeah, because the winch is on the E-gen. So
Yep, another Analopsamia rostrata, and the white part is not alive, and the yellow part is. You can see the polyps there in the calyces of the scleractinian coral. And then we have a shrimp just behind it, and a sponge, glass sponge just in front. Yes, one. I thought it was. I think it was 3.
Alright, video good, come wide.
Hmm.
Some lasers on it. Do we get lasers real quick? Perfect. Alright, do a little push forward. Back there. It's
yeah.
kind of behind us.
I'm
Video.
still
Looks
at
like a
60%,
Thuarella over
so
to the right that we just passed. And then we have a little
Kinda
bit
steep,
more
Kinda see if it tapers at
Selenosmilia.
all.
this is 90.
That's 90. Okay.
Pilots, could we zoom on this hard coral that's right on the left? Yeah, and the yellow coral as well. Thank you.
Yep. Alright, video could come in.
Yep, definitely Selenosmilia. Ooh, and that's a Chrysogorgid, I believe, on the left of this Selenosmilia, and it has its associate shrimp. So that Chrysogorgid is growing on the
You come
dead
partial real
part
quick?
of the
Try to
Selenosmilia.
get a little closer.
And then we have a Stichopathes black coral right next to that in the center. And then we're going to take a look at this yellow coral, which my guess is that that's a skeleton that of a previous coral, and these are zoanthids that are growing over
Our video could come in
the
for yellow.
former coral that has now. Yeah, those look like zoanthids to me. And from previous experience, these zoanthids actually bioluminesce. So if you were to bring them up on the ship and give them a shake, you would have this beautiful coloration, bright blue bioluminescence.
go ahead, Survey.
And this was probably a bamboo coral that that was the original former of the structure
Yes.
that the zoanthids are now growing over.
Yes.
And you can see a glass sponge just to the left of this that's
no, we have a
also no
Spec
longer
7,
alive.
a biological in the port inner. Yep.
Alright, video, whenever you're good, we can rotate back right.
Do you want to take over here?
There's 2 bios in the port outer. They're
We'll
both
come
in the
back
same
in
box.
on orange.
So yeah, spec 1 and 2
Or
are
I
in the
guess-
port outer.
Sorry, Washley, did you say something?
Okay.
Yeah, intentional. Yep.
Wow, so that was another little oasis of life here.
All right.
And you can see lots of areas of dead scleractinian coral skeleton. It makes up a lot of what we're seeing in this area here. And that usually means that as we go up slope, we're going to find a lot more alive, and the dead parts just kind of roll down this steep slope over time and get colonized by other organisms.
Copy. Move complete. I can get you going if you need. Where are you at? No, he's not quite yet.
Hold on that.
ROV nav bridge, move complete.
Move complete. Thank you, Bridge.
Something's moving in there.
Yeah, video go zoom. I think fish. Dead center. Yeah, there he is.
Pilot, this is Watch.
Go ahead, Wash.
Yeah, I heard what you're saying. Yeah, let's take a look at this fish and look around this rock that's covered with stuff.
Bridge, this is RV nav.
This is Bridge, over.
Yeah, how do the forces look on your DP screen? I see my indicated current on my systems kind of show that it's kind of on your starboard beam, but it's pretty low, 0.2. Seeing how you guys are at, what you guys see.
Yeah, it's pretty light today. It is absolutely beautiful up here. We have the wind directly on the bow and the current just slightly on the starboard bow, but the wind is the overriding factor right now.
Okay. Thank you very much.
Video, let's let them float out. Actually, it's going to stop. All right. It's all right. Video com wide. Let's take a look at what else we have here. Copilot, you can grab lasers. Great.
What about these? Oh, those two are Stauropathes, and then what about this biggest one? Is that also? Oh, okay. So 3 Stauropathes with the Stichopathes in the center. Oh, look at them there. There's got some really big ones off to the right.
It's a cool shelf.
Yeah.
It looks nice.
It's really angular. It's like plates piled up, or like, you know.
Thanks. Yeah, so that is a Liopathes, the big orange coral that you see there. It's yet another species of black coral. So I don't know, for up to 5 or 6 species of black coral on this dive alone. And there's only 16 black coral species known from this area. So we have really seen quite a diversity. Yeah, it's amazing. That's also a very beautiful sponge over here.
Is that the scleractinian coral?
Do you know how to route it over there?
That's, I don't know how to pronounce that.
Selenismilia variabilis.
Let me just do it.
No, it's called ROV still, I think.
And look at this sponge on the left here,
too.
Oh,
I wonder if that's similar to the satellite disc one that we collected the other day.
It looks so much thinner, though. That one was quite thick, and there were also veins that you could see in the Phycalia, and kind of.
Some of those pictures.
Actually, it's some more Selenismilia underneath that sponge, the white one on the left. Gosh, this is a beautiful spot.
Awesome.
Does it?
We have a brittle star on
right
an octocoral
so
that's hanging straight down,
looking
also
at
off
the
of
sea
this
star,
ledge.
the brittle star on the bottom.
What about this
the bottom? So if you raise it a little
coral
bit,
in front of
turn
this
to starboard.
yellow sponge?
Yeah, I'm
Do you know
not quite sure about that yet.
Pilot, this is watch.
Yeah, watch. Go ahead.
Pilot, this is watch 2.
Watch. Can you hear me?
Yeah, sorry, I messed up on the
No,
button
you're good.
there.
How can I help?
Can we zoom in on this coral that's just in front of the yellow sponge?
Absolutely.
Thank you.
All right, video, go for zoom.
That looks again like kind of like an Acanella, but I don't think I'm right on that, but similar.
I was actually thinking Anthophila, but
looks like possibly a little cup coral or an anemone on the right of it too, be kind of behind.
Yeah, there's also a couple of polychaete tube worms here. Yeah, Candidella. It's a primnoid, is what Scott Francis suggested.
Okay.
So that's one we haven't seen before. Oh, it got
a hermit crab on the top of that.
I don't know if that's actually on the sponge or the coral, and a crinoid on the behind the coral, Candidella. And we have the Selenosmilia on the right side, the stony coral. Beautiful yellow glass sponge. So it looks like there's some some kind of
associates in the sponge.
That's looking good, Sean.
Know if those are scale worms, probably.
Oh, looks like another type of glass sponge behind the yellow glass sponge, with some crinoids in it. Lots of hydroids all over the place.
All right, watch leads. Is that good?
Yes, if you could zoom out and continue to explore this area, there's quite a few species here we want to just document.
Copy that.
If you rise a little bit, like you'll get the whole shot on the still cam.
Copy. Video come wide. Real quick. Let's see what we got. And then yeah, come by.
It is amazing.
Yeah, just come rise a little bit.
A little more.
Yeah. Like you see it right there?
Yeah.
How it's looking.
Pilots, there's an octocoral that's hanging straight down,
right,
and it has a brittle starfish, a reddish brittle starfish on it. Could we take a look at that, please?
Yeah. It's down below, correct?
Yes.
Yep.
All right, I think we got a few pictures that were really good.
Nice, thank you, co-pilot.
I'll turn off the camera. And then video, do you want me to leave those lights on, off, how? Sorry, I can't hear you. Hold on.
There's quite an abundance of life on this
Video, go for zoom. Is this the one, watch it?
Yes, I believe it is. Cheryl just stepped out for lunch, so.
All right.
Stephanie, was this the one that?
I'm not sure. Yes.
Annie on our video team just thumbs up.
Yeah, we're doing musical chairs right now, so I was didn't exactly hear what she was saying, but thankfully Annie's paying attention.
Okay, now we're settled.
All right, video, whenever you think you're got what you need. Come back, we get lasers real quick so we can measure. Nice.
Stephanie, did you want to look at the sponge that was on the left, or had we already looked at that? That large sponge.
Do you want these lights on?
The disc-shaped fan one. We didn't take a close look at it. Maybe that would be good.
Sorry, Washley, what was the next?
Pilot on this watch. Sorry.
Yeah, if we can zoom out, and to the left is a large white disc kind of sponge hanging at the edge.
Yeah, it's
is it
down,
in view?
Down a little bit from
actually,
this rock. It's the next rock down.
It's hanging from the next balcony.
Copy that. We back up. Pirouette copy. We
And
can go
to
back
the left
to
of this. Should be right there, yeah.
Okay, so let me know when you want me to kill lasers.
So I was asking Cheryl if she thought this was
quick.
The same or similar to the
And
ones we were seeing many of the Facalia we were seeing the other day, and she thought it was not, but maybe some of the sponge experts can weigh in.
Yeah, it does seem thinner, and she mentioned it has those veins.
Yeah, it does look different.
Has the same idea, though, doesn't it?
Yes.
Facing into that current, and it's sitting, well, it's actually pushed up against the
navigator. What's the highest
like
point?
It's growing, coral. Yeah, it's growing over some of that scleractinian coral.
1260.
Okay.
Whose name I'm not going to try to pronounce.
I'm going to try because I wrote it out phonetically.
Looks like I got a 1250.
Selenus melia.
That's the thing. I need to see it before I could say it. Anyway. So like I said, we're playing musical chairs right now because it's the lunch. So just to remind the people out there, this is Stephanie Bush from the Smithsonian Institution National Museum of Natural History. And
I'm Leslie Sauter from the College of Charleston Geology Department. And before I forget, I want to also say a shout out to my team back on campus in the Visualizations Lab. Trey Gillespie and Sophie Alpert are there today, and I know Zach Pro, my grad student, is also sitting in with them.
Video.
Thanks, Pilot.
With that? Great.
They're hosting tours if you're in Charleston.
Video, let's do that pirouette.
Seeing some great overhanging rock slabs.
Let's start.
Pilot, this is Watch.
Yeah, go ahead, Watch.
So let's just you started going kind of right, and just let's keep going in that direction. Just survey what's around this rock area.
So keep going right?
Yes, please.
Copy that.
Unless you had something.
Oh, do you want me to circle it?
No, the pirouette. Or
I think
they want
to keep.
I think they don't want it now, right?
That's a great question.
Yeah,
we're thinking.
Oh,
Hold on a second.
Since I was absent, I'm not quite sure where we are. Maybe we can pull away a little bit and get a view of the area.
Back up.
That'd be great. Thanks.
Oh, sorry. I thought. Copy. I wasn't aware where it was coming from. That was the confusion.
We see a crinoid up there on the right. We've zoomed into those a few times.
Lasers on.
And then, yeah, watch these. We have been here for a little bit. So if there isn't anything super interesting you'd like to zoom on,
yeah,
probably should move on.
I'd like to keep climbing if we could.
Yeah, let's keep going.
Great.
Copy that.
Maybe I should move in.
Yeah, absolutely.
Thank you, Pilot. That was some great flying on these biota.
No problem. You're very welcome.
I mean, 280 is good with me.
Okay. 280.
Bridge, this is ROV nav.
Well, I would call this a rocky mess.
Is that an appropriate geological term?
Yeah, it is now. Wow, that's...
Bridge,
this is quite a wall here. And you can see these rock slabs have fallen from above. So if we grabbed them, we don't really know where their origin was at this point. Do you want Stephanie, shout out if you see anything you want to take a zoom in.
Pilot, this is Watch.
Yeah, go ahead.
Can we kind of slow down and take a look at these corals that are basically just at the bottom of the center of the screen?
Copy that.
Thank you.
Bridge, this is ROV now.
Video, go ahead. Zoom in on these.
So we've got the Stichopathes right here. Pilot, this is Watch.
Go ahead.
Can we take a look at the sort of lighter pink one? Yeah, that. Oh.
You got it.
Thank you.
Kill lasers.
This looks similar to one of the corals we saw in that previous rock. I can't remember the name, and I actually got logged out of the chat room. But I know that Scott had a name for it. Oh, Candidella? Is this individual also that species? Or
Bridge, this is ROV now.
This is actually
yeah, we'd like to move 20 meters, bearing 280 degrees, speed decimal 2.
Bamboo coral.
Okay, 20 meters.
Yeah, we need to get a little bit of a closer look if we want to go any further than that, but the overall appearance of it.
Pilot's is Watch.
Color,
yeah, Scott was just asking for a closer look on this, so got it, thanks.
The polyps and the skeleton and the thickness suggest to me it might be the jasonitis clade.
Video I can try to set if we need a really good close-up. I'm just kind of floating right now above other coral.
Move initiated. Thank you, Bridge. All right, pods, I got a move going here.
Copy that. Copy.
Yeah, we're at
is that good, or do we need better footage of that? I can try to set down.
This is okay. We can move on. It's definitely a new bamboo coral for the day.
All right. You can come wide then. Left. Copy.
And this orangish coral looks to me like what we've been seeing at Stauropathes, but I would not be surprised to be wrong on that.
No, I agree, Stephanie. That is correct. Stauropathes, black coral.
Great. I'm learning. This is good.
One of the values of participating in these dives is how much we all learn from each other.
Very true.
I want you to know I'm trying.
Copy that. All right, watches. Is that good for this area?
I'm delayed here, so you probably passed it, but there's also a small brittle star.
Yeah, I did see that. We really kind of have to keep going, but we did see that. Sorry, we didn't point it out for everyone.
Yeah, what's interesting about that, it may have been Okeanos is the genus name. And what's interesting is you don't see that type of brittle star on the corals. They're completely different. So that tells us something about the evolution of some groups of ophiuroids as associates with the corals and other groups that are independent.
Do they just roam around on the rocks instead, Scott?
I'm sorry, Leslie, could you repeat that?
Do they roam around more on the rocks and just scavenge, or?
Right, exactly.
Yeah.
They seem to be scavengers that... the way I would interpret it, they're either not fishing out of the water column, and so many of these ophiuroids are climbing up on the sponges of the coral simply to get into the faster-moving water, so they're using it as kind of like a tree to hang off of a tree. But then there are others that are maybe feeding on the coral, feeding on the mucus, or feeding on things that land on the coral or sponge. So there's all kinds of different strategies.
150 meters.
Copy.
So it's interesting that, we're seeing these slabs that are not horizontal, and we don't see nearly the growth on them, and yet we do see sedimentation on them. So they've been in this position for a while, so this does not seem to be prime habitat for these species we're finding in abundance on some of the slabs that are a little more horizontal. We saw a great balcony earlier, just about 10 minutes ago, and it was high density of organisms all. cohabitating.
Yeah, it was good diversity of corals and sponges, and then all the things that live on them, in them, among them as habitat. Pilot, this is Watch.
Yeah, go ahead.
Can we take actually there's a white, I think, sponge that's just going off the bottom center. Can we take a look at that?
Copy that.
Seems to be another one on the rocks, almost dead center, if that's
yeah,
the same
either
thing.
one, they look similar, so whichever one is easier for us to zoom in on.
Copy that. I'm starting to make my way here, so we'll go for bottom one. Just give me 1 sec, video. Get level to it.
Looks like a glass sponge to me, but I don't remember seeing ones like this earlier today, or so far today. Could be wrong on that. Oh, also, just to point out, on the left, there's a break in the rocks, and there's some crinoids hanging down in there.
Move complete.
To the left, we've got this. We just zoomed past it, but there was a bathypathes. Oh, there's an anthomastus down on the lower left.
So this is sponge and coral.
Sponge and coral, yeah. So a glass sponge with some coral.
Zoanthid. It looks like a white zoanthid is overgrowing a glass sponge. And if you give me a minute, I'll try to find out the approximate grouping that this sponge belongs to.
Okay, great, Scott. Thanks.
And that's another of the bamboo corals to its right. I think the same as we were just imaging a few minutes ago.
Okay, like, you mean the Canada... Oh, wait. I forget what name you said on that. Sorry.
I'll lift for a little bit, see if it passes. Maybe we'll just make our way to
this. Looks like another bathypathes to the right, as well as more of that scleractinian coral. Seleno...
Feel any current, Sean? Or?
Leslie can pronounce it for us because she has it written down.
Like
Selenosmelia.
Selenosmelia. I think that's looks like a larger,
mostly
dead colony, but with some living tissue on the top.
It does clear pretty quick, actually.
There's a greenish sponge. It looks like the same
type
of sponge, just
but
a different color.
Yeah.
Do you want to use zooms? Flashlights?
That'd be great. Thank you.
Was it dead center?
Yes.
Copy. Video go in.
Or actually the greenish one that was slightly right of center.
Slightly right, copy.
And up. Little.
And up, yeah, sorry. Not center of screen. Yeah. It's in the upper left now of the screen.
Oh, upper left.
Yeah.
Is it tucked back in there?
Mm-hmm. Yeah, it's a greenish color. Maybe that's...
It does look similar shape-wise, but
part of it looks clear.
Yeah, it really does.
Huh. Changes color.
Beautiful. And it looks like there's possibly a scale worm on the top of it. On the rock in front of that sponge, you can see lots of branching hydroids and a few encrusting tunicates. That's great, Pilot. Thanks.
Right. We pan left, maybe just a little bit. There was, yeah.
Yeah, that looks like another Stauropathes, possibly.
And a crinoid is living under the rock to the left.
Which doesn't strike me as a good place to be if you want particulates and coming across in the water column, but
all right, video come wide. Back it up here.
All right, should we
keep going
on?
Keep going.
Yeah.
Copy that.